Hold up your right hand. Now bend your thumb backward, away from your palm, as far as it will comfortably go. Watch what happens at the last joint, the one closest to the tip. For some people, that joint barely moves at all. The thumb stays almost straight, maybe curving back a few degrees before it stops. For other people, something different happens. The tip of the thumb folds backward until it is almost lying flat against the back of the hand, forming a sharp bend that looks like it should not be physically possible. If your thumb does that second thing, you are carrying a trait that people have been noticing, joking about, and misunderstanding for well over 100 years. It has a nickname almost everyone has heard: Hitchhiker's thumb. And for most of the last century, people believed they understood exactly where it came from and exactly how it was passed down through families. It turns out almost everything about that old explanation was wrong. The real story, the one that modern genetics has only recently been able to piece together, is stranger, older, and far more interesting than the simple rule taught in classrooms for decades. Before we get into what the newer research actually found, take a second to subscribe to this channel if you like this kind of slow, detailed look at the small physical details written into the human body. There is a lot more of this coming, and subscribing is what tells the platform that this kind of video is worth showing to more people who would enjoy it. Turn on notifications too, so you catch the next one when it goes up. Now, back to your thumb, and to a scientific mystery that took most of a century to properly untangle. People noticed thumb hyperextension long before anyone had a name for it. Old family photographs sometimes show relatives holding up a bent thumb as a party trick. Traveling performers in the 19th century sometimes included it in acts alongside other displays of unusual flexibility. Doctors examining patients in the early 20th century wrote it down as an interesting but harmless variation, the same way they might note an unusual ear shape or a widow's peak in the hairline. Nobody thought much of it beyond curiosity. It was a party trick, a talking point, something that ran in certain families and skipped others for no obvious reason. The first real scientific attempt to explain the trait came in the 1950s and 1960s, when human genetics was still a very young field. Researchers at the time were fascinated by traits that seemed to follow clean, simple patterns of inheritance, the kind Gregor Mendel had described in pea plants a century earlier. Earlobe attachment, tongue rolling, and thumb flexibility all became popular examples in that era, taught in biology classrooms as illustrations of dominant and recessive genes. The story told about the flexible thumb was simple and tidy. A single gene, it was claimed, controlled the trait. A straight, rigid thumb was said to be dominant. A hyperflexible thumb was said to be recessive, meaning a person needed 2 copies of the gene, one from each parent, to show the bent version. Textbooks reproduced this explanation for decades. Millions of students were taught to check their own thumbs and their parents' thumbs as a simple lesson in genetics, tracing a trait that seemed to move cleanly from one generation to the next, according to basic rules. Interest in the trait was not limited to biology classrooms. Long before anyone had run a genetic study, thumb hyperextension had already made its way into folk medicine, into circus and sideshow performances, and into casual family lore passed down at dinner tables. Grandparents would show grandchildren that their thumbs bent the same dramatic way, and the family would treat it as proof of a shared bloodline, a little physical signature passed down the generations like a nose shape or a hairline. In some communities, it picked up superstitious meanings entirely unconnected to genetics, treated as a sign of luck, of a particular temperament, or simply as a curiosity worth pointing out at gatherings. None of these folk explanations had any real scientific grounding, but they show how long people had been paying attention to this small detail of the hand long before anyone had the tools to explain where it actually came from. The problem was that the rule never actually held up under careful testing. As more researchers began measuring larger and more diverse groups of families through the 1970s and into the 1980s, the clean pattern kept breaking down. Children showed thumb flexibility that did not match what the simple recessive model predicted. Identical twins, who share essentially all of their DNA, sometimes showed different degrees of thumb bend from each other. Populations studied around the world showed a wide, continuous range of flexibility rather than 2 neat categories of flexible and rigid. Some people had thumbs that curved back only slightly. Others had thumbs that bent almost 90 degrees. Many people fell somewhere in between, showing partial flexibility that did not fit into either box the old classroom model demanded. By the late 20th century, most geneticists had quietly abandoned the single gene explanation, even though it kept circulating in textbooks and trivia lists long after the underlying research had moved on. What replaced the old model was something more complicated, and honestly more true to how most human traits actually work. Thumb hyperextension is not controlled by one gene switching on or off. It is influenced by many genes working together, each contributing a small amount to how loose or how tight the connective tissue is around the joint at the base of the thumbnail. This kind of trait is called polygenic, meaning many genes, and it produces exactly the kind of smooth, continuous range of outcomes that researchers were actually seeing in their measurements. Instead of a light switch, think of it more like a dimmer switch controlled by dozens of small hands at once, each nudging the settings slightly higher or slightly lower. The genes involved turned out to center heavily on the body's connective tissue, the material that holds joints, skin, and internal structures together, and gives them both strength and flexibility. The most important family of genes in this story codes for collagen, the main structural protein found in tendons, ligaments, cartilage, and skin. Collagen genes such as COL1A1, COL3A1, and COL5A1 were identified as major contributors to general joint flexibility across the whole body, not just the thumb. Small variations in how these genes are built change how densely packed and how tightly cross-linked the collagen fibers end up being. Looser, more elastic collagen allows joints to bend farther before the surrounding ligaments stop the movement. Tighter, more rigid collagen restricts the range of motion and produces the straighter, less flexible thumb that most people have. Alongside the collagen genes, researchers found contributions from genes involved in elastin production, a different structural protein that gives tissue its ability to stretch and spring back into shape, and from genes that regulate the enzymes responsible for breaking down and remodeling connective tissue over time. All of these systems interact. A person's final degree of thumb flexibility depends on the combined output of dozens of these small genetic variations working together, plus a smaller contribution from how the joint develops physically in the womb and in early childhood. This is why the trait runs loosely in families without following the clean rules of classic Mendelian inheritance. Parents pass down a mixture of many variants, not one clean switch, and the mixture their children inherit can land anywhere along the spectrum. Once researchers accepted that the trait was polygenic and continuous rather than a simple dominant recessive pair, a new question opened up. If the trait is influenced by dozens of small genetic variants scattered across the genome, are those variants evenly distributed across every human population on Earth? Or do some populations carry certain variants at higher frequencies than others? This is exactly the kind of question that large-scale genetic studies have become very good at answering over the past 15 years, thanks to genome-wide association studies, often called GWAS, which scan the DNA of tens of thousands of people at once and look for patterns that line up with a measured trait. Several research groups working on general joint hypermobility, which includes thumb hyperextension as one of its most visible and easiest to measure signs, ran exactly this kind of large-scale genetic screening during the 2010s and into the 2020s. They recruited large cohorts, measured joint flexibility using standardized scoring systems that doctors already use to assess hypermobility, and then compared the genetic data against the measurements. What emerged was a clearer picture of which collagen and connective tissue variants were most strongly associated with greater flexibility, and where in the world those variants tend to show up more often. The population data told an interesting story. Broad hypermobility surveys conducted across different countries and different ethnic groups over the past several decades had already noticed something worth explaining. Average flexibility scores were not identical everywhere. Populations in parts of South Asia, the Middle East, and West Africa tended, on average, to score somewhat higher on standard joint hypermobility scales than populations in parts of Northern Europe and East Asia, although the differences were modest and there was enormous overlap between every group measured. These were never sharp dividing lines. They were shifts in the average, the kind of pattern you only notice clearly when you measure thousands of people and run the statistics, not something you could ever guess by looking at any single hand. But the pattern kept appearing, study after study, decade after decade, which told researchers that something real and at least partly heritable was sitting underneath the numbers. When the genetic variant data was laid on top of these population patterns, several of the collagen and connective tissue variants most strongly linked to greater joint flexibility turned out to sit at higher frequencies in exactly the regions where the older hypermobility surveys had already flagged higher average scores. This did not prove a simple one-to-one relationship because dozens of variants are involved in environment, nutrition, and general health also affect how joints present later in life. But it gave researchers a genetic explanation that lined up with decades of earlier physical measurements, which is exactly the kind of convergence that makes a scientific finding feel solid rather than coincidental. The deeper question, the one that pulled researchers into the field of ancient DNA, was where these variants came from in the first place and how long they had been traveling with human populations. Ancient DNA research has transformed the study of human history over roughly the past 15 years. By extracting and sequencing genetic material from skeletal remains, sometimes many thousands of years old, scientists can compare the DNA of people living today against the DNA of the ancestors who actually walked across specific regions of the world in deep prehistory. This lets researchers trace when a particular genetic variant first appears in the archaeological record and how its frequency shifted as populations moved, mixed, and settled into different environments over tens of thousands of years. Applying this approach to the connective tissue variants linked to joint flexibility showed that many of them are old, some of them very old. Versions of the collagen gene variants associated with looser, more flexible joints appear in ancient genomes recovered from populations that lived in the region encompassing modern-day South Asia and the Arabian Peninsula many thousands of years before written history began. They also appear, at somewhat different frequencies, in ancient remains recovered from parts of West and Central Africa. These variants did not spring up recently. They were already circulating in human populations deep in prehistory, carried along as those populations expanded, split, and moved into new territories across the millennia. As with so many traits shaped by ancient population movement, geography and climate may have played a quiet role in how these variants spread and settled at different frequencies. Some researchers have proposed that connective tissue flexibility carries subtle advantages in certain physical contexts, potentially related to childbirth, certain kinds of manual labor, or general physical adaptability, though this remains an area of ongoing debate rather than settled fact. Others point to simple genetic drift, the random rise and fall of variant frequencies that happens naturally in any population over long stretches of time, especially during periods when a population was small or was expanding rapidly into new territory. Most researchers in the field currently favor some mixture of both explanations, drift setting the initial pattern and mild selection pressures reinforcing it in certain environments over many generations. Nobody claims to have the full answer yet, and that honest uncertainty is part of what makes this an active area of research rather than a closed case. What all of this means is that if you have a thumb that bends back sharply, the genetic variants sitting behind that flexibility carry a documented history that, in many cases, traces back through extremely old branches of the human family tree, through populations that lived in South Asia, the Arabian Peninsula, and parts of Africa long before recorded history, and outward from there into every region those populations eventually reached through migration. This does not mean that everyone with a bendy thumb has ancestry from these specific regions. The relevant variants exist in every population on Earth at some frequency. Plenty of people with Northern European ancestry, East Asian ancestry, Indigenous American ancestry, and every other background on the planet have highly flexible thumbs. The trait is universal in its distribution. What differs between populations is not whether the variants exist, but roughly how common they are. And that difference in frequency is what produces the population-level averages researchers have been measuring in hypermobility studies for decades. For people who already know their family history includes ancestry from South Asia, the Arabian Peninsula, or West and Central Africa, this genetic research offers a small, quiet piece of context that earlier generations never had access to. The flexibility in your thumb is not simply a random accident of your individual development. It is, at least in part, a visible trace of connective tissue variants that have been present in your ancestral lineage for an extraordinarily long time, carried forward through thousands of generations of people you will never know the names of, across enormous stretches of geography and time. For people whose family history traces through Southeast Asia, the story carries an extra layer of complexity, because that region has long served as a genetic crossroads. Multiple ancient population waves moved through and settled across Southeast Asia over tens of thousands of years, some arriving very early in the human expansion out of Africa, others arriving later alongside the spread of agriculture and new language families. Each of these waves carried its own mixture of connective tissue variants and modern populations across the Philippines, Indonesia, Vietnam, Thailand, and Malaysia typically carry genetic contributions from several of these ancestral streams layered on top of each other. The hand cannot tell you which layer contributed which fraction, but the layering itself is part of the story written into the DNA. For people of West African, Central African, and East African descent, the picture also involves real regional variation, since the African continent holds by far the deepest and most genetically diverse human population history of anywhere on Earth. Populations that stayed in different regions of Africa across tens of thousands of years developed distinct genetic profiles long before any migration out of the continent took place, and the frequency of connective tissue flexibility variants shifts noticeably from one African region to another. Large-scale hypermobility surveys conducted in West African countries have repeatedly recorded some of the highest average flexibility scores found anywhere in the world, a pattern that lines up with the elevated frequency of several key collagen variants identified in more recent genetic screening of West African cohorts. It is worth pausing here to address what this trait does not mean, because there has been no shortage of exaggerated claims made about thumb hyperextension over the years, especially online. A flexible thumb is not a sign of athletic talent, and it is not a reliable predictor of skill in music, sports, or any other physical pursuit. Despite persistent folk claims connecting hypermobility to talents like playing string instruments or excelling at gymnastics, it is not connected to intelligence, personality, or any measure of general health in the vast majority of people who have it. For the large majority of the population, a bendy thumb is simply a harmless variation, no different in its practical impact than the shape of your earlobe or the whorl pattern of your fingerprint. In a smaller number of cases, significant joint hypermobility across multiple joints in the body, not just the thumb, can be one sign among several that a doctor might consider when evaluating a person for a connective tissue condition such as hypermobile Ehlers-Danlos syndrome. But a flexible thumb on its own with no other symptoms is not something to be concerned about, and the overwhelming majority of people with bendy thumbs never experience any joint problems related to it at all. Reading too much into the trait in either direction, treating it as either a hidden talent or a hidden health warning, misses what the careful research has actually shown. What the science has actually found is more modest, and in its own way more interesting than either exaggeration. Most people go their whole lives thinking about their thumb flexibility only as a curiosity, something to demonstrate at a party or compare with a sibling. But once you understand what the flexibility is actually recording, the joint becomes something closer to a small, quiet archive, sitting at the base of your thumbnail, carried with you everywhere, checked without a second thought every time you grip something or make a fist. Thanks to the combined work of geneticists, physicians studying joint hypermobility, and researchers working with ancient DNA, that quiet archive can now be read with far more understanding than any earlier generation had available. If you look at your thumb again with this in mind, you might notice it a little differently than before. The angle at which it bends, whether sharp or shallow, was shaped by a specific combination of collagen and connective tissue variants inherited from both of your parents, variants that were themselves inherited from generations stretching back far beyond memory, far beyond written history, into populations that were adapting to environments and building lives in specific corners of the world tens of thousands of years before anyone alive today was born. The thumb is a small joint. The story sitting underneath it is not small at all. There's one more layer worth mentioning because it brings the whole picture together in a way that connects this research to tools many people have direct access to today. As consumer genetic ancestry testing has become widely available over the past decade, some smaller research groups have started asking whether measured thumb and joint flexibility correlates at all with the ancestry percentages those tests report. Early results suggest a weak but noticeable relationship. People whose test results show a higher percentage of ancestry from South Asia, the Arabian Peninsula, or West and Central Africa do tend, on average, to report slightly higher joint flexibility scores than people whose ancestry results point mainly toward Northern Europe or East Asia. The relationship is nowhere near strong enough to use as a stand-in for an actual ancestry test, and nobody serious in the field would ever suggest using thumb flexibility that way. But across large groups of people, the pattern holds up, and it adds one more piece of evidence connecting the genetic story to the population averages that hypermobility researchers have been documenting in clinics and physical exams for decades. This kind of research keeps expanding every year as more genomes, both ancient and modern, get sequenced and compared. Twin studies added another useful layer to this picture during the same period that the genetic screening work was underway. By comparing identical twins, who share nearly all of their DNA, against fraternal twins, who share only about half, researchers could estimate how much of the variation in joint flexibility comes from genetics at all, separate from environment or lifestyle. These studies consistently found that a large share of the variation in thumb and general joint flexibility, generally estimated somewhere in the range of half to two-thirds depending on the specific study and the joints measured, could be attributed to inherited genetic factors. The remaining variation came down to a mix of age, activity level, connective tissue changes that happen naturally over a lifetime, and simple measurement differences between one exam and another. This estimate lined up well with the polygenic model that the genome-wide association studies were independently arriving at, giving researchers two very different kinds of evidence, one from family comparisons and one from direct DNA analysis, pointing toward the same conclusion from two separate directions. For anyone who happens to have a highly flexible thumb, this can be a small moment to think about how directly connected your present-day body is to an extremely deep and distant past. You did not choose the specific mixture of collagen and connective tissue variants sitting in your DNA. You did not choose the ancestors who carried those variants across deserts, coastlines, river valleys, and mountain ranges over tens of thousands of years, long before any modern nation, language, or culture existed in anything close to its current form. But the inheritance is real, and it is sitting quietly in one of the smallest joints in your body, doing something it has been doing in human hands for an extraordinarily long time. There's also something worth appreciating in how universal this trait remains, even with its regional patterns of frequency. The relevant variants exist in every human population studied so far. The trait itself shows up in every human group on Earth. What differs is never presence versus absence, only how common the variants are in one region compared to another. When researchers map joint hypermobility across the globe, they do not find sharp borders separating one population from the next. They find a smooth, gradual shift in averages from one region to another, with enormous overlap at every point along the way. Two people, picked at random from any two different populations on Earth, are far more likely to resemble each other in thumb flexibility than to differ sharply because of where their ancestors happen to live. The pattern belongs to populations, expressed only as an average across thousands of measurements. It was never meant to describe, and can never accurately describe, any single person's hand on its own. So, the next time you catch yourself bending your thumb back, whether to show a friend or just out of habit, you're looking at more than a strange little party trick. You are looking at a small physical record of connective tissue genetics passed down through an unbroken chain of ancestors, some of whom lived and moved across the earth so long ago that the very idea of a country, a border, or a written language did not yet exist. The joint is small. The history behind it is not.